the body's universal fuel into actual energy.
And yeah, this is far from just basic chemistry. We've got a pretty dense stack of sources today. They take us way beyond the textbook stuff. We're talking sophisticated regulations, some surprising clinical problems, and even metabolic warfare when things go sideways. Think environmental toxins, even cancer.
Right. So our mission here is to move past just the simple steps and really understand the complex implications, like how does glycolysis actually dictate oxygen transport or hormone responses, and why is it such a critical choke point in how our cells make energy?
Exactly. The foundational importance of glycolysis is that it gives cells ATP, that's the energy currency, whether or not oxygen is actually around. Normally, glucose gets broken down and fully oxidized to CO2 and water. That's, aerobic oxidation.
Okay, but what if oxygen is scarce? What happens then?
Well, then the process stops short. It converts the glucose into two molecules of lactate instead. That's anaerobic glycolysis. And we see this anaerobic reliance all over the place, muscle cells during really intense exercise, tissues starved of oxygen, and famously in your red blood cells.
Right. Red blood cells, they completely depend on anaerobic glycolysis because they don't have mitochondria for the aerobic part. But what I find really fascinating is how other cells, ones that do have working mitochondria, sometimes choose to lean into this anaerobic process anyway.
And that leads us perfectly into discussing the really tightly regulated steps. Glycolysis isn't just an open tap. It's got three key irreversible reactions. They act like gatekeepers, making sure the cell only really commits to burning glucose when it absolutely needs to.
Okay, so let's start with the very first one, phosphorylating glucose, right? That traps it inside the cell. And we've got two main enzymes doing this job, hexone kinase and glucokinase. What's the functional difference there?
Well, they're isoenzymes. Same job, basically, but built for different situations, different tissues. Think of hexokinase, or HK, as the sort of cautious one. You find it in most tissues. HK has a really high affinity for glucose, grabs it even when levels are low, but crucially, it has a low capacity, a low Vmax.
So it gets full easily. It puts up the stop sign pretty quickly.
Precisely. HK is inhibited by its own product, glucose 6-phosphate. As soon as the cell has enough of that, HK just stops. Now, compare that to glucokinase, GK, that's in the liver and the pancreatic beta cells. GK has a low affinity, but a huge capacity. It just keeps going.
Right, that huge capacity must be vital for the liver, especially dealing with that massive flood of glucose right after, say, a big sugary meal.
It's exactly that. It's the liver's way of being the body's glucose sort of sponge or floodgate. GK's high Vmax lets the liver pull that sugar out of the blood fast, minimizing hyperglycemia after you chug a soda.
And here's that aha moment. Our sources really highlight GK isn't just an enzyme in the pancreas, is it? It's doing more.
Yeah, it actually functions as a glucose sensor in those pancreatic beta cells, works alongside the GLUT2 transporter there. Its activity basically tells the pancreas how much insulin to release and how quickly. And it acts as a sensor in the brain too, in the hypothalamus, signaling low glucose.
Wow. And when that really sensitive glucokinase sensor is mutated, well, that's when we see a specific, often kind of quiet disease pop up. MODY2, maturity onset diabetes of the young type 2.
That's it. MODY2 is caused by a mutation right in that pancreatic glucokinase gene. The sensor doesn't work quite right, so the body consistently makes just a bit too little insulin. It can be really subtle, sometimes totally asymptomatic. Just shows how much our whole body glucose balance depends on that one single enzyme working properly.
Okay, so we've trapped the glucose. Now we hit the second irreversible step, our critical.
Because PFK1 catalyzes the committed step. Once you go past PFK1, that glucose molecule is definitely going all the way down to pyruvate. There's no turning back, really. And it's incredibly sensitive to the cell's energy status. If ATP levels are high, the cell's got plenty of energy, right? So PFK1 gets shut down by ATP and also by citrate, another energy-rich signal. But if energy is low, shown by high AMP levels in peripheral tissues, PFK1 gets switched on.
And the liver has its own special accelerator for PFK1 linked to hormones.
It does, yes. It's regulated by this clever enzyme system called PFK2-FB-PASE2. This system makes a molecule called fructose 2-pel-6-bisphosphate. When you've eaten, insulin activates the system, boosting fructose 2-pel-6-bisphosphate. And that molecule is a super powerful activator of PFK1. Then when you're fasting, glucagon flips the switch, reverses this, and shuts down glycolysis in the liver, stops it from burning precious glucose.
It's just amazing how many layers of control are built in. But this complexity also means vulnerabilities, right? Especially to toxins. I was pretty shocked reading about arsenate.
Oh, arsenate is nasty. A real metabolic saboteur. It works through chemical mimicry. See, there's a crucial step in glycolysis catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It needs inorganic phosphate to make a high-energy intermediate. Arsenate, which you can get from contaminated water or soil, looks chemically almost identical to phosphate.
So it basically tricks the enzyme.
Right.
But what happens then?
It competes for that phosphate binding site. Glycolysis looks like it's continuing, but the arsenate intermediate that forms is unstable. It just immediately breaks down, hydrolyzes, and the consequences, well, it's devastating. The cell completely skips the step where it makes the first high energy phosphate bond, and crucially, it doesn't generate any NADH at that step either.
Wait, hang on. If it skips that high energy step,
yeah,
what does that do to the overall energy count, the ATP?
It drops the net ATP yield from one glucose molecule completely to 0. You still use 2 ATPs at the start, right? But now you only make 2 ATPs back at the very end at the pyruvate kinase step, that first substrate level phosphorylation step. Gone, bypassed. So for cells that really rely heavily on glycolysis, like maybe in low oxygen conditions, arsenate toxicity means catastrophic energy failure.
Wow, that is stark. Okay, on a much, much lighter note, but still common, fluoride.
Yeah.
I genuinely never realized fluoride preventing cavities was a metabolic thing.
Yeah, it's actually a pretty neat bit of biochemistry. Fluoride acts as a non-competitive inhibitor for the enzyme enolase. That's one of the later steps in glycolysis. The bacteria in your mouth that cause cavities, they need glycolysis to survive and make acid. So inhibiting enolase basically starves them. And here's a little clinical aside. That same inhibition is why they add fluoride to blood collection tubes when they want to measure your glucose levels accurately. It stops the red blood cells in the tube from gobbling up the glucose before the lab can measure it.
That's actually brilliant. Okay, let's jump to the third and final irreversible reaction, the one catalyzed by pyruvate kinase, or PK. This step gives us the enzyme deficiency, right?
That's right, pyruvate kinase deficiency. It's inherited recessively. And remember, red blood cells rely entirely on glycolysis for their ATP. So if their PK enzyme doesn't work properly, they just can't make those final vital ATP molecules.
And without ATP, the cell's basic maintenance fails, like the pumps that keep ions balanced.
Exactly that. The NAP plus McK plus pump, the sodium-potassium pump, is critical. It uses ATP constantly to maintain the right osmotic balance inside the red cell. When that pump fails due to lack of ATP, the cell swells up with water, gets deformed, and eventually just bursts. That's hemolysis. Clinically, this shows up as mild to moderate hemolytic anemia.
But even with this problem, the body has this weird sort of compensatory trick linked right back into glycolysis itself.
It's really fascinating how the body finds these workarounds. It involves something called the Rappaport-Lubbering shunt, specific to red blood cells. Because the main pathways block downstream at pyruvate kinase, the intermediates before that step start to build up. Specifically, 1,3-bisphosphoglycerate accumulates. This shunt pathway takes that 1,3-BPG and converts it into a different molecule, 2,3-bisphosphoglycerate, or 2003-BPG.
Okay, why is that molecule 2,3-BPG helpful?
Because 2003-BPG is a major player in oxygen transport. High levels of 2003-BPG bind to hemoglobin and make it release its oxygen more readily to the tissues. It causes what we call a right shift in the oxygen-hemoglobin dissociation curve. So strangely enough, the high 2003-BPG you see in PK deficiency actually helps with oxygen delivery, somewhat offsetting the anemia.
It's amazing. Okay, let's shift gears now from specialized cells like red blood cells to, well, cells that are growing like crazy cancer cells. We have to talk about the Warburg effect. This whole phenomenon where cancer cells just gorge on glucose and spit out lactate, even when there's plenty of oxygen around. I mean, doesn't that seem incredibly inefficient, wasteful even?
It does seem counterintuitive, absolutely, because aerobic respiration yields so much more ATP per glucose molecule. But Otto Warburg noticed this decades ago. Most cancer cells preferentially do aerobic glycolysis. They choose to make lactate even with perfectly functional mitochondria sitting right there. It turns out it's less about raw ATP yield and more about speed and building materials.
Speed and building materials. So why is making lactate so fast actually good for a tumor?
Well, there are a couple of key reasons emerging from the research. First, all that lactate gets pumped out of the cell. This makes the area around the tumor really acidic. And that acidic microenvironment seems to promote angiogenesis, the growth of new blood vessels to feed the tumor, and also helps the tumor invade nearby tissues. It sort of melts the surroundings. Second, by shunting glucose towards lactate quickly, the cell keeps the upstream glycolytic intermediates readily available. It needs those intermediates as building blocks for making new lipids, nucleotides for DNA, amino acids, everything needed for rapid growth and division. They prioritize building over just pure energy efficiency.
That makes a kind of sinister sense. Now, speaking of lactate buildup, what pushes things from a normal temporary lactate increase, like after you sprint, into dangerous pathological lactic acidosis?
Right, lactic acidosis is serious stuff. We're talking blood lactate levels climbing above, say, 5 millimolar, which then drags the blood pH down below 7.2. That's metabolic acidosis. This usually happens when there's a critical imbalance between NAD plus and NADH, the electron carriers. The most common triggers are severe problems getting oxygen to tissues, like in shock or major trauma or septic shock. Also, defects anywhere along the electron transport chain in the mitochondria can cause it.
And our sources also flagged heavy alcohol consumption as a big factor here. Why does booze stress the lactate system so much?
Yeah, alcohol metabolism puts a huge strain on the liver's NAD plus supply. Breaking down ethanol requires a ton of NAD plus brain, converting it all to NADH. Now remember, you need NAD plus to convert lactate back to pyruvate, which is how the body normally clears lactate. If all your NAD plus is tied up dealing with alcohol, pyruvate just gets forced down the path to lactate instead. It piles up fast, leading to acidosis.
Man, glycolysis really is central, isn't it? It connects to everything. Before we wrap up, maybe let's just quickly list some of the key pathways that branch off or depend on these glycolytic intermediates.
Oh, absolutely. They feed into almost everything important. You've got glycogen synthesis storing glucose for later, triacylglycerol synthesis making fats, synthesizing several non-essential amino acids, and crucially, the pentose phosphate pathway branches off early. That's vital for making ribose sugars for DNA and RNA, and also NADPH, which the cell needs for antioxidant defense and building fatty acids. It's a real metabolic hub.
Okay, perfect. So to kind of bring this whole deep dive together, let's just quickly recap the energy yield, the ATP situation, what are the different scenarios we talked about?
Sure. So standard aerobic oxidation of 1 glucose gives you a net of 2 ATP directly from glycolysis, plus the reducing power from 2 NADH molecules, which yield much more ATP later in the mitochondria. Anaerobic glycolysis going all the way to lactate, or if you have defective mitochondria, gives you just that net 2 ATP from glycolysis itself. And the really stark one, under arsenate toxicity, because it sabotages that key step, the net ATP yield from glucose plummets all the way down to 0.
That's a fantastic summary. It really drives home that glycolysis isn't just some static flow chart. It's dynamic. It's regulated by hormones, by oxygen, and yet it's surprisingly vulnerable to these outside attackers.
Exactly. And we've talked a lot about what happens to glucose inside the cell. But remember those 2 NADH molecules generated out in the cytoplasm during aerobic glycolysis? To get the maximum energy out of that glucose, the energy stored in that cytoplasmic NADH needs to somehow get into the mitochondria where the big ATP payoff happens.
Right. Which brings up a really interesting point, something for you listeners to maybe chew on after this.
Yeah.
Does every tissue in your body actually squeeze out the exact same amount of energy from every single molecule of glucose?
Yeah, the answer lies in these things called shuttle mechanisms. The cell uses specific transporters to get that NADH energy across the mitochondrial membrane. There are 2 main ones. If a cell uses the glycerol 3-phosphate shuttle, that cytoplasmic NADH effectively yields about 1.5 ATP's worth of energy inside the mitochondria because it passes electrons to FAD. But if the cell uses the more efficient malate aspartate shuttle, that same cytoplasmic NADH yields a higher payoff, about 2.5 ATP's worth, because it passes electrons directly to NAD plus inside.
Wow, so a difference of a whole ATP per NADH,
Yeah.
That's a huge difference in energy efficiency, and it just depends on which shuttle system your particular tissue happens to use. Something to think about. Definitely food for thought. Well, thanks for diving deep with us again today. Keep digging into those details.